Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the Deep Dive. This is where we break down complex material, find the absolute highest yield nuggets of knowledge, and, well, deliver them straight to you as an intellectual shortcut. And today's mission, it's a big one. We are tackling a full summary of NCERT Class 11 biology. This stuff is foundational, and we're looking at it through a very specific lens, efficiency. For anyone targeting competitive exams, especially if you're zeroing in on the UPSC. But before we dive in, I really want to take a moment, a huge heartfelt shout out to all of our existing subscribers and members. Your support, it honestly makes it possible for us to put in the hours needed to synthesize this dense material. So thank you. And if you're listening and you find this kind of focused analysis useful, please consider helping us out. Subscribe, hit that like button, share a comment, and if you're in position to, joining the paid membership is how we keep this content coming.
It is a massive subject. I mean, we're covering everything from the simplest cell to, complex human physiology. And our goal today isn't just to list facts, it's to show you why certain facts become high-yield questions. We're going to distill the critical concepts linking the NCERT text to that broader knowledge you need.
Okay, let's unpack this, starting with the fundamental definition of life itself. So the sources list things like growth, reproduction, sensing the environment. But, a crystal can grow. So for a really precise, exam-ready definition, what are the two core properties that truly define a living organism?
That distinction is, it's absolutely paramount. For something to be universally defined as living, we really have to look at two things: metabolism and cellular organization. Metabolism is just the sum of thousands of chemical reactions happening inside the organism. If you do those in a test tube, you have living reactions, sure, but you don't have a living thing.
So the container matters. The structure that holds all those reactions is the key.
Precisely. That's the second one. Cellular organization. The cell is the non-negotiable unit. You look at growth or reproduction, and they have exceptions. Mule can't reproduce, but it's very much alive. Metabolism and organization, they're mandatory.
And what about consciousness, that ability to sense and respond?
Consciousness is also a defining property, definitely, from a simple microbe all the way up to us. But the source material makes a really interesting and specific point here. Only one organism, the human being, has self-consciousness, an awareness of self. That's a key distinction they love to test.
Okay, let's talk about the simplest life forms, prokaryotes.
Right.
Bacteria. They lack organelles, but their classification is crucial. Why is that classification by Shapetoccus, Bacillus, Vibrio spirillum still so fundamental?
Because morphology, especially at that microscopic level, it's a constant. It's reliable. You just have to remember those four basic shapes because they're the bedrock of identification. Spherical is coccus, rod-shaped is Bacillus, comma-shaped is Vibrio, and spiral is spirillum. Those names just need to be locked in.
And inside, the ribosome size gives us this beautiful structural clue. We have 70S and 80S types. Where do we find them?
This is one of my favorite parts of cell biology. The smaller 70S ribosomes, they're characteristic of all prokaryotes. But here's the fascinating part. You also find them inside two eukaryotic organelles, the chloroplasts and the mitochondria.
Which is the core evidence for the theory that these organelles were once their own separate organisms, right?
Exactly. The endosymbiotic theory in a nutshell. It suggests they were engulfed by a larger cell billions of years ago. The larger 80S ribosomes are the ones you find floating around in the main eukaryotic cytoplasm.
Moving up to biochemistry now, metabolites. Why do we even bother making a distinction between primary and secondary metabolites?
It's all about their role and how widespread they are. Primary metabolites are your universal building blocks, amino acids, sugars, things you find everywhere because they're essential for basic growth. Secondary metabolites, on the other hand, are more specialized. They're unique to certain groups: plants, fungi, microbes. Think alkaloids like caffeine or rubber or essential oils.
So the caffeine isn't essential for the coffee plant's basic survival, but it acts as a defense mechanism, a secondary function.
That's a perfect way to put it. They're often ecologically or medically vital, even if they're not part of that core growth pathway.
And one last thing for this section, size. How do we draw the line between a micro and a macromolecule?
It's basically a molecular weight cutoff. The big ones: proteins, nucleic acids, polysaccharides. These macromolecules are usually in the range of 10,000 Daltons and above. So when you do an acid extraction in the lab, these huge molecules precipitate out. They form the acid insoluble fraction, literally separating themselves by size.
Okay, let's switch gears. We're going from the microscopic cell to the whole plant, specifically morphology of flowering plants. This chapter is just a goldmine for questions because of all the adaptations. Let's start with leaves. How does a leaf modify itself to do something completely different from photosynthesis?
Oh, the adaptability is incredible. It's all about survival. For climbing, leaves can become thin tendrils like you see in peas. For defense, they sharpen into spines, which you see on cacti. For storage, they get thick and fleshy, like an onion or garlic.
Right. And the Australian acacia is a fantastic example of a functional shift.
It's a great one. In the Australian acacia, the real leaf is tiny and falls off early. What you think is the leaf is actually the petiole, the leaf stalk. It expands, turns green, and completely takes over the job of photosynthesis. It's just functional flexibility.
That's a great detail. Okay, now flower arrangement, or inflorescence. This can get confusing, but the difference between racemose and cymose always comes up. What's the core difference in their growth?
It really just boils down to what happens to the main growing tip. In a racemose inflorescence, the main axis just keeps growing. It never ends in a flower. Because of that, the youngest flowers are always at the very top. We call this acropetal succession.
So new flowers appear at the apex, growing upwards.
Exactly. Now, in the cymose type, the main axis does end in a flower, so its growth is limited. The new flowers then have to develop from buds below it. This is called basipetal order, meaning the oldest flower is at the top. The key is knowing acropetal versus basipetal.
Let's connect that structure to the plant's circulatory system. What's the one single rule to remember about transport direction in xylem versus phloem?
It's the difference between a one-way street and a dynamic two-way highway. Xylem moves water and minerals. That always comes from the ground up. So the flow is strictly unidirectional, from the roots up.
A one-way trip?
Always. But phloem transports food, which is mostly sugars made in the leaves, and the plant's needs change. So phloem transport is bidirectional. It moves sugars from a source, like a leaf, to a sink, like a growing fruit or a root. And what counts as a source or a sink can change, so the flow has to be flexible.
That dynamic flow is a perfect lead-in to essential elements. Let's talk nitrogen. When it comes to biofertilizers, which specific cyanobacteria do our sources highlight as being key, free-living nitrogen fixers?
The two exam favorites are always nostoc and Anabaena. Both are cyanobacteria. They're crucial because they're free-living and can pull nitrogen straight out of the atmosphere and convert it into a form plants can use. Just knowing those two names is high yield.
And the actual process, nitrogen fixation, has a weakness. Oxygen. How do plants, specifically in root nodules, protect the vital enzyme that does this job?
Right. The enzyme is called Nitrogenase, and it's completely inactivated by oxygen. So the whole process has to be anaerobic. The solution in legumes is incredible. They produce a compound called Leghaemoglobin. It acts as an oxygen scavenger, binding up any free oxygen to protect the enzyme and let it work.
That is just a fascinating bit of biochemistry. Now, the source material also had some interesting cross-disciplinary facts. What's the link between biology and oceanography when it comes to fishing grounds?
Yes, this is a great general science point. The most important fishing grounds in the world are found where warm and cold oceanic currents meet. That mixing of waters causes a massive upwelling of nutrients, which creates these biological hotspots.
And from a geography perspective, what's the defining feature of equatorial forests?
Their single most unique characteristic is the presence of tall, closely set trees with crowns that form a continuous canopy. This dense ceiling of leaves defines the whole ecosystem by blocking out most of the light.
Back to plant physiology for a moment. Deficiency. What's the scientific term for the point at which a lack of an element starts to hurt the plant?
We call that the critical concentration. It's the concentration of an essential element below which the plant's growth is noticeably held back. It's the threshold for deficiency.
And finally, water absorption. Water gets into the root through two main pathways. Can you contrast the Apoplast and Symplast routes for us?
Yeah, think of it this way. The Apoplast pathway is like water taking the highway. It moves through the non-living spaces, the cell walls. It's fast and continuous until it gets blocked by the Casparian strip.
And the Symplast?
The simplest pathway is water taking the local streets. It actually enters the living part of the cell, the cytoplasm, and moves from cell to cell through little bridges called plasmodesmata. It's slower, but it gives the plant much more control.
All right, let's pivot to human physiology. Starting with the heart's own pacemaker. What is it, and what kind of rhythm does it set?
The main controller is the sinoatrial node, or SN. We called it the pacemaker because it has this innate ability to generate action potentials, initiating them at a rate of about 70 to 75 per minute. That sets the rhythm for the entire heart.
And that rhythm creates a measurable constant. What's the standard duration of one complete cardiac cycle?
One full cycle atria and ventricles contracting and relaxing takes a very precise 0.8 seconds. That's one of those physiological constants that you just have to know.
And just quickly for transfusions, which blood group is known as the universal recipient?
That would be the AB blood group. They're universal recipients because their plasma doesn't have A or B antibodies, so they won't attack red blood cells with either A or B antigens.
Okay, on to the body's electrical system. The neuron. It maintains that resting state positive outside, negative inside through a really energy-intensive process.
Yes, that resting potential is actively maintained by the sodium-potassium pump. This is critical. It's always working, pumping 3 sodium ions out for every 2 potassium ions it brings in.
So it's deliberately creating an imbalance, moving more positive charge out than in.
Exactly. That 3 for 2 trade is what creates the net negative charge inside the cell. It establishes the electrical gradient, getting the neuron ready to fire.
And when it does fire, the action potential, what's the immediate chemical event that flips the charge and causes that wave of depolarization?
The signal is a sudden, massive change. An impulse triggers channels to open, allowing a huge, rapid influx of sodium ions. This rush of positive charge into the cell is what reverses the polarity and sends that depolarization wave down the nerve.
We've covered a ton of biology, but as promised, the source also had some great general science facts. For our last nugget, what common phenomena are actually, according to physics, just optical illusions?
This is a great one. 2 common things we see are pure optical illusions. First, the exaggerated size of the sun at dusk when it's on the horizon. And second, the familiar twinkle of stars in the sky. That's just caused by our own atmosphere bending the starlight. They aren't actually twinkling.
It's all just perceptual effects, not changes in the actual objects. Amazing.
That was a really great synthesis. I mean, we covered a lot of high-yield ground. Defining life, differentiating metabolites, the rules of plant transport, and then nailing down constants like the 0.8-second cardiac cycle.
It really is incredible how all this science connects. But here's the final thought I want to leave you with, and it brings us right back to where we started. We established that the cell is the definitive feature of life. So think about viruses. They have genetic material, but they can't metabolize or reproduce on their own. How do these simple borderline things challenge our very neat textbook definitions of living? It's a great question to think about. And once again, thank you for diving deep with us. We are so grateful for the support of our existing members who make this all possible. If you found this deep dive valuable, please subscribe, like the video, share your comments, and if you're able, becoming a paid member is the most direct way to support us so we can keep bringing you these focused summaries. We'll catch you on the next deep dive.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.